All right, good afternoon everyone. Thank you for the opportunity to present our work today here, and also thanks to AR for Soil Health, where we're part of the project, which is very great. So today I want to tell you a bit more about our device that measures extracellular enzymatic activity, and this is what we do. And so you know this is not just an idea, it actually exists. This is Bob. Bob flew in this morning from Stockholm. And today I want to tell you a bit more what we measure, why we measure, and how we measure. So enzymatic activity is a biological soil indicator. And if you look at soil indicators, they're physical, chemical, and biological ones. But happens to be that very often if researchers measure a minimal data set, biological indicators are underrepresented. And reasons are manifold, but very often it's like there's very few indicators that are widely accepted among the community, but also that the spatial and temporal resolution is really bad, which is often because it's difficult to measure or very expensive. And if we look at biological indicators, the soil functions that you look at with them is either biodiversity, nutrient cycling, or filtering, for example. And what we want to look at is especially nutrient cycling. And I don't want to go too much into detail, but this is an example of cycling of nitrogen in the soil-plant system. And, well, very easily said, so you have an input of organic matter, for example, by fertilizer, where nutrients are bound. And in order to make these nutrients available for a plant again, you need a couple of processes, and all the arrows, especially the blue ones, are ones where enzymes are involved. So enzymatic activity is a very nice indicator to look at nutrient cycling. But what actually are enzymes? So enzymes are proteins. And those proteins, they act as a catalyst. So they're biological catalysts. And what they do is you have big molecules, and the enzymes cleave certain bonds and make the molecules smaller until a point where they're small enough for organisms and plants to take it up again. And when we talk about enzymes, we talk about a substrate. Substrate is the organic molecule. We have the enzyme, and in this example, the enzyme facilitates the cleavage of a bond with the help of water. That's a hydrolytic enzyme. And what you have in the end is a product, so two smaller molecules. So the work of enzymes is basically decomposition, depolymerizing large molecules to smaller ones. And most of the enzymes in soil actually derive from bacteria, but all living organisms produce them. Also, you have them in your stomach when you eat. Basically, it's the same principle. You eat, and in your body, the enzymes free the nutrients so you can take them up again. So when we look at the very example, the very nice thing about enzymes is they're substrate specific. What do I mean with substrate specific? It means that a certain enzyme just cleaves a very certain bond. So we have the example here of the phosphatase. On top you have a phosphate molecule where organic rest is bonded, and the phosphatase only cuts this certain bond, the phosphomonoesterase. So we know when we measure the activity of phosphomonoesterase, we know a phosphate ion has been released. That's what we mean with substrate specific. We really can trace a very certain process. And another thing that is very important to know is we measure extracellular enzymatic activity. So you can have enzymes in cells, and you can have enzymes out of cells. And most organisms, like roots also, but also microorganisms, they excrete proteins to the outside. So at this point, the proteins are also subject to abiotic interactions like sorption or degradation due to temperature or frost. So what extracellular enzymes are kind of subjected to abiotic and biotic processes, and they integrate over both, which makes them very interesting. So now you know what we measure and you know why we measure, but the question is, how do we measure enzyme activity? So we do this with fluorescence. So there are certain molecules, if you excite them with light of a certain wavelength, they emit fluorescence. So, for example, So, I can use this. This is quinine, but be sure we don't measure with tonic water. But the principle is similar. So we use artificial substrate. So it's a molecule, like they happen to be present in soil, but they have a dye attached that is fluorescent. And once the enzyme cuts this bond, fluorescence is emitted. So the more bonds are cut from this artificial substrate by the enzyme themselves, the more fluorescence is emitted. And this is what we measure. And this relationship of substrate and activity is known as the Michaelis-Menten kinetics. And what we measure is the release rate of a number of molecules per soil sample per time. And this has been researched by this guy and this girl, Michaelis and Maud. And what basically is very important to know is the more substrate you give, the more food you give, the more fluorescence will be released. But at a certain point, you have a limited number of enzymes. If they run throughput, like full throughput, you can give more food, but they won't produce more fluorescence. And this is where we want to measure, which is the plateau on top, the Vmax. So what we need to ensure to get to Vmax is that the substrate is not limiting for the measurement. For this, and to make up our setup here, we did a couple of experiments with different soils. And on the left, you see on the x-axis, we have different concentrations of substrate. And we measured for certain soils the enzymatic activity. And you see at the beginning, it goes up. The more food you give, the more fluorescence you have. Then there's a plateau. But if you give too much, just quenching, it goes down again. This is how we set the substrate concentration. And how does this actually look like in our real setup? What we have is a reaction plate with different compartments. And in the green ones, we have substrate of the defined concentration. And actually, we have 5 different substrates in an analytical replication of 3. So we can measure, for now, 5 different enzymes at once. And then we have in blue a calibration well, so later we can translate the fluorescence that we measure in the substrate wells into actual number of molecules from the calibration. How does this look in real life? So you have your soil, you put it in a little box, you take the reaction plate that holds the chemicals in a gel, you put this on top of the soil, you put it in a bowl, and you press start, and then it measures. And what you get out of this is a data set that consists of photos that document the increase of fluorescence over time. From there, we extract the luminosity values, we convert them with the standard curve in concentrations, we take the linear part of the concentration increase over time, and we get a rate of extracellular enzymatic activity, and this is all implemented in an automated data analysis pipeline. And what we currently measure are 5 different enzymatic activities from the nitrogen, the carbon, and the phosphorus cycle. And we will probably implement more enzymes that, depending on your needs, what is available. So this setup can do many more than just these. So you might think now, well, nice box, nice concept, but who tells me that this actually works? And this is why we conducted a couple of validation experiments. And the first one we did was very simple in a way. We bought enzymes, and we spiked a sand with different concentrations of enzymes, and in this spiked sand, we measured enzymatic activity. And you can see here one example. So if you increase the concentration of enzymes, we get a linear response in the increase in enzymatic activity that we measure with BOP, which is basically a proof that it does what it should do. But there's everyone who is in soil and everyone who is a bit in analytics knows there's many influencing parameters. And there's a couple of that influence the measurement, and there's a couple of that influence enzymatic activity. And you need to be aware of these. So one basic thing that influences enzymatic activity is the amount of substrate. So we talked about this before. So we make it non-limiting. So this is a fixed parameter. And the other is the number of enzymes, but this is an inherent property of your soil. in a certain time period. So we make sure that we measure in a time period that the number of enzymes does not change. Another thing that's very influencing is soil texture and organic carbon content, because this can quench fluorescence. And in order to make sure that it works for very different soils, we have a set of soils with a very broad range of texture, but also a very broad range of organic carbon content, to make sure that we also are able to measure across all the soils, or most of the soils that you probably can bring to us. The next thing is pH. pH is very important for enzymes because enzymes have an optimum pH value where they work best. And if you deviate from this pH, there can be conformational changes in the protein, and that, for example, the active site. And that makes them less active. So what we did, we tested also the soils of a pH for now 4 to 8 to make sure that we don't run into problems of pH with a shifting spectra or shifting activity. And then we decided to measure at soil pH, but it would be also possible to put a buffer and you can decide at which pH you want to measure. Similar to pH, we do have temperature. So there's the optimum temperature. If you go closer to this from the left, you have more activity because of kinetics. If you cross this, at a certain point you go to denaturation. And what we did, and my co-founder wanted to kill me for this, I put the whole device in the oven and in the fridge. So we have a range of 6 degrees until 55 degrees. And we measured for different soils the activity at different temperatures. And well, first, the devices survived, all of them, promise. And second, we see the typical increase, and then at the 55 degrees at the plateau that we would expect. So you can measure at different temperatures with this device, and it responds in a way that we would expect it to respond. The last point I want to address is water content. So water content also affects enzymatic activity a lot, because mainly you need to bring the enzyme and the substrate together, otherwise they cannot react together, right? So that is something that happens in soil. If it's dry, there's less happening. There's like no decomposing in your compost, for example, if it's too dry. So you need moisture. What we have in our setup, the gel in the reaction plate contains water. So even in dry soil, this facilitates minimum contact in order to measure. But it doesn't swamp the whole sample. And we did with 8 different soils and 5 different soil moisture content experiments. There's no pattern. It depended a bit on the soil type, which kind of pattern we got, but this is also that other people found in literature that soil water content has no unified pattern of the effects of soil moisture, but we could show that we can measure from very dry to pretty moist samples. And this was just a very short insight, so we submitted a publication with all this, so if you want more detail, I hope it won't take too long until this is published. And with this, I'm coming to the end of my presentation, and what I want you to take home is next time you drink your gin tonic, I want you to remember that it has amazing molecules that can fluoresce. If you put UV light on it, unfortunately, I was not allowed to open the bottles here on stage and drink, but maybe we can do this later. But what I really want you to take home is that we have this device. It's pretty easy to use. It can be used anywhere, and we tested it for a wide range of soils and environmental conditions in a very standardized way. And maybe this makes it suitable for using it more to have a better temporal and spatial distribution as a biological soil indicator. And this will be Charlie that we will produce next year and will be on the market. And thank you for listening.